Hello, I'm Samantha Richard, and I partnered with Michelle Valkanas to investigate soil fungi for antibiotic properties and characterization. Antibiotic resistance is becoming a huge problem as our available antibiotics are becoming less effective in treating infections. This is because antibiotics continue to be overprescribed and insufficiently regulated. As a result, a selection pressure is generated driving pathogenic organisms to mutate and share resistant genes. The discovery of new antibiotics is crucial in combating drug-resistant infections. In our study, fungi was derived from a soil sample and tested for potential antibiotic production. Antibiotics commonly come from fungi and bacteria present in the soil. We also characterize them through microscopy, staining, and phytochemical analysis. We hypothesized that antibiotic producers would be discovered in our study, as there is an abundant amount of fungi present in the soil, which would result in a wide array of differing cellular mechanisms. We began our study by collecting 5 grams of soil near Monongahela River in California, PA. 6 serial dilutions were performed, and the last 4 were plated in duplicate on Sabouraud Dextrose Agar medium infused with streptomycin. After incubation, fungi were picked and patched based on characteristic morphology on SD agar plates with streptomycin. These became our master plates. We then repatched them on SD agar without streptomycin in preparation for our antibiotic screen. Our antibiotic screen was then conducted using Escherichia coli, Staphylococcus epidermidis, and Pseudomonas aeruginosa. We performed the screen by spreading the tester strain on nutrient agar plates and transferring an agar plug of each isolate onto each tester strain lawn. All of the isolates were observed under a dissecting microscope for hyphae structure. They were also stained with cotton blue and observed under a compound light microscope. We performed this by mounting a fragment of the isolate onto a slide with water and adding the stain. Fungal crude extract was also prepared for each positive isolate by inoculating them in SD broth, filtering the broth, and resuspending the residue after evaporation with DMSO. Each extract was used for phytochemical analysis to see if they were endophytic fungi. If they were, this would help other researchers to target them for antibiotic production, and it would also show how the fungi relates to the plants and their symbiotic relationship. Each extract was tested for tannins, coumarins, quinones, and flavonoids by adding a certain chemical reagent and observing a color change. 11 unique fungal isolates were derived from our dilution plates. Isolates 1 through 4 and 6 were similar in color. Isolates 5 and 7 through 9 were found most unique in their color, shape, and texture, while isolates 10 and 11 had similar cottony top growth, but were different in their color underneath. After 3 days of incubation, our screen produced 4 positive isolates due to the presence of a clearing. Isolates 4, 6, 10, and 11. Isolate 11 had the largest clearing of about 1.5 millimeters. Interestingly, these isolates were only effective against S. epidermidis, a gram-positive bacterium. For timely reasons, I only showed the positive isolates under a dissecting microscope and compound light microscope after staining. Isolate 4 and 6 continue to be similar even in their hyphae structure, though in the stain, isolate 6 has black nodules presumed to be spores, though this could be due to a longer incubation time with the release of spores. Isolate 10 and 11 have differing hyphae structures, so they may be related, but they might not be the same species. Our phytochemical tests only produced one positive result, which was for coumarins. Coumarins are present in higher plants and other microorganisms. They serve as a defense mechanism against herbivores and other microorganisms. This result was a weak positive from isolate 4, so we can't necessarily characterize them as an endophytic fungus. It might just be a soil-based fungus. The purpose of our study was to investigate fungi derived from soil for antibiotic production. We also characterized them through microscopy, staining, and phytochemical analysis. Our antibiotic screen produced 4 positive isolates, which further supports our hypothesis, though our other isolates could have antibiotic properties since we only tested them against 3 pathogenic bacterial strains. The characterizations we made helped recognize differences between isolates and how they could play a role in how effective they are at inhibiting the growth of pathogens. Further studies to continue this research could be identifying positive isolates to recognize their novelty or analyzing the compound responsible for their antibiotic activity. This study further supports the need of a worldwide initiative. Even though we found antibiotic producers, we don't know if they've already been discovered yet, though this study validates that soil is still a valid place to look for new antibiotics. Without new antibiotics, microorganisms will continue to mutate and share their resistant genes, decreasing the effectiveness of our available antibiotics. I'd like to thank Michelle Valkanas for assisting me through the research process and procedures of this study, as well as the Department of Biology, Geology, and Environmental Sciences for providing the resources to successfully perform this study. Thank you.